Hey, this is Miss Ramon. This video is going to be our review for the test that's coming up. The week 7 test, which covers chapters 12 and 13 from your textbook. The people upstairs are having a party and my dogs keep trying to bark, but we're making this video anyway because I'm in charge here. All right. Chapter 12. This covers the cell cycle and mitosis. And as we've talked about in class, there is a lot of vocabulary in this unit, in both of these chapters. So, and then there are a lot of terms that begin with the letter C, which makes things even more difficult. So spend some extra time learning the vocab. Quizlets are good, flashcards, friends that you trust. You can quiz each other because nobody wants to get the question wrong because they misread what the word was. All right, so DNA. We are going to describe and discuss DNA and using a lot of different terms. And those terms can describe structure, location, a time period, or stage of the cell cycle. So I tried to section this off with those terms that you're most likely to confuse with each other. So the term molecule, you may not think you could call something as huge as DNA a molecule, but it is. DNA is a molecule. The double-stranded DNA that's anti-parallel to each other, that's hydrogen bonded to each other, that is a molecule of DNA. The hydrogen bonding between the two strands counts as a bond, so it is a unit, and that makes it a molecule. So anybody who tells you that hydrogen bonding is not significant, you can tell them no, don't yell at them. I mean, don't bark. Good girl. Okay, sorry. People upstairs are having a lot more fun on a Friday night than me making this video. I'm just kidding. Of course, I love doing this for y'all. Okay, so molecule of DNA, when you hear that term, that's the double-stranded DNA with the double helix. Chromatin is a term that's used to describe all of the DNA and chromosomes that's in the nucleus. And it's usually referring to a time when the cell is just living its cell life. In the G1 stage, it's just being a cell and doing what it does. And if you were to look at that cell under the microscope and stain for the DNA, you would not be able to see individual chromosomes. It would just be very kind of grainy. So that's the chromatin. And the chromatin is made up of all of the individual chromosomes. The term chromosome is one molecule of DNA with all of the proteins that are associated with DNA when it's inside of a cell. So molecules of DNA aren't just lying around. They are wrapped around proteins that are called histones. So a chromosome means the DNA molecule and all of the proteins that it is wrapped around. And chromatid is a term that's only used after the DNA has been replicated. So once the DNA replicates, and you have two identical copies of a chromosome, they are attached together by their centromeres, and that entity, that duplicated chromosome, is a singular chromosome because they are attached at their centromeres, and then you would call those identical copies sister chromatids. So as soon as the DNA is duplicated, the two identical copies are bound together, and that one entity is a duplicated chromosome, a single chromosome made up of two identical sister chromatids. And when the sister chromatids separate from each other, they are considered daughter chromosomes or just chromosomes. A chromatid only means something when there are sister chromatids. They don't exist by themselves. When they're by themselves, they are considered a chromosome. And here are some more images from the Khan Academy website. They have a lot of really good images. And if you look at this top picture on the left, you see where it's labeled DNA. So here's that double-stranded, double helix structure that you're used to seeing. And then for the chromosomes, you notice there are these histone proteins. And the DNA wraps around the histone proteins. And then those histone proteins with the DNA can also, they can clump together and then spiral and then pack together really, really, really tight. And then we get this X structure that you're used to seeing in textbooks. And this is sort of that classic X structure that we think of when we think of chromosome. And what this actually is a condensed chromosome. You can't see uncondensed chromosomes. That's why we call all of the uncondensed chromosomes together chromatin, because we can't tell the difference between any of them. They're not really visible with a light microscope unless they're condensed. And when they condense, the histone proteins are going to be a big part of that in helping it pack very, very tightly into that X structure. And once again, the chromatin in the cell, when the cell's living the cell's life, it looks like chromatin, even though all the chromosomes are there. When it's time for the cell to divide, the cell gets a signal to go through the S phase and DNA replication occurs. And at this point, we now have sister chromatids connected to each other by the centromeres. And then they start to condense before they enter into mitosis, which is that separation process of the nucleus. Because if you tried to separate these unwound, very, very, very long pieces of DNA, they would break. So any organism that used to do that did not live long enough to have children to pass on that trait. Only those organisms that packed their DNA tightly survived. And so that would be the reason why this happens. All right, the cell cycle. This is another image I got actually from Wikipedia. And I really like this image because they just packed everything you want to know about the cell cycle and mitosis and cytokinesis. So, good for them. That's pretty awesome. But it is a really good image, I think, because it has everything you need to know. Plus it has pictures of what the stages of mitosis look like. And it's really not difficult to read. So that's quite impressive. The only thing is, and it took me a moment to notice, and I put an extra arrow in here, is that this is going counterclockwise. So I highlighted in yellow those 3 stages of the cell cycle. So the cell cycle has 3 stages, interphase, mitosis, and cytokinesis. Interphase has 3 stages, G1, S, and G2. Mitosis has 4 stages, prophase, metaphase, anaphase, and telophase. So there's a lot of stages that have stages, that have stages, that have stages, and then they have traits. And you do need to know all of those, and that's just purely factual information. Once again, Quizlet might be a great way to learn all of that. And really, there's nothing in here that I need to give you any extra information on. Let me just double check. Yeah, this is a great overview picture, I think. Okay, now we're going to look a little closer at the stages so that I can point out anything you need to pay special attention to, especially anything that might end up as an application question. So in Interphase, the three stages are the G1, S, and G2. And remember that G1 is when the cell is doing its job. Nope, no. Okay, she's gonna bark. That's, Joko, no, I think it was a bird. It was not a monster. It was not a monster. Okay. No, actually, I think that's another dog. I think that's another dog. All right, sorry, I had to take a break because my dog was barking to protect me. Something was outside, and then I'm explaining to my dog that, it's really hard to work from home during a pandemic. I don't have much choice. All right. So Interphase has three stages, G1, S, and G2. And G1 is the time period that the cell is just doing its normal cell job. You will see the nucleolus. They have this green dot here that's supposed to be the nucleolus. And then you see that the chromosomes in the nucleus are not condensed. And this is normally what chromatin refers to, is when the DNA is not condensed, because when the cell is just living its cell life, it needs to produce proteins because that's how the cell is able to do its job. So the DNA needs to be accessible for all of the machinery that will convert some of these messages into proteins. Things that you should notice: the nucleolus will be visible as a dot. Chromatin is the term we use to describe all the chromosomes. Notice there is only one centrosome, and the centrosome is composed of two centrioles that are these barrel shapes that are 90 degrees, that are at 90-degree angles to each other. During the S phase, there has been some kind of cell signal telling the cell that it's going to need to divide, and that's the only reason it would go into the S phase. If it's not going to divide, it doesn't go into the S phase. So if it gets the signal to go in the S phase, then a second copy of the chromosomes are made, and then those identical copies are bound together by the centromere. And then this new chromosome is composed of two identical sister chromatids. Even though the mass of the DNA has doubled, we still have the same number of chromosomes. It's just that the chromosomes have an identical copy and they're bound together by the centromere, and that counts. That counts for them actually staying one chromosome. All right, then G2, during this stage, there are final preparations for the cell to divide. Notice we now have two centrosomes, and the nucleolus is still visible. The nuclear membrane is still intact, and we have duplicated DNA molecules, or the chromosomes are duplicated, or the chromosomes have two identical sister chromatids bound together by the centromere. There's lots of different ways you can say it. But what's important is we have 2 centrosomes now. We did not have 2 centrosomes during G1. The nucleolus is still there. That's important. The nuclear membrane is still there. That's important. And the reason that's important is because that will change during mitosis. And I put a list of questions that you should think about every time you look at the mitotic phases, because these would be the type of questions that either I would ask you, or the ACT would ask you, or the AP exam, or dual credit, or the college course that you take in a couple years. These are just classic questions for this material. So being able to determine if the cell is diploid or haploid. Another way to ask that would be, are there homologous pairs present or not? Is the chromosome duplicated or unduplicated? And that's also referred to as replicated and unreplicated. So that will mean those, you might see those used interchangeably. Another question is, where are the centrosomes? Where are they located in the cell? They're going to be moving around. Is the nucleolus still visible? Is the nuclear membrane intact? You could also be asked, what is the chromosome number of a cell? What is the value for N? And are the chromosomes condensed or uncondensed or not condensed? These are all typical questions. Okay, so mitosis begins with prophase. And in early prophase, we have the 2 centrosomes that are starting to move to the opposite ends of the cell. The nuclear envelope is starting to fall apart, and the nucleolus is not visible anymore. Late prophase. In late prophase, the centrioles or the centrosomes are going to be at the poles. They have to be at the poles so that metaphase can occur, because they are going to put the chromosomes in the metaphase plate. So the centrioles and the centrosomes must be in place themselves before they can put the chromosomes in where they're supposed to go. And the way that the centrosomes are in charge of getting the chromosomes where they need to be is by the spindle fibers. So the spindle fibers are also cytoskeletal elements that are going to be, they're going to extend from the centrosomes, and the spindle fibers actually are being built and being broken down at different rates so that it extends the spindle fibers out, and then they latch on to the kinetochore. They're like little hooks, and they hook onto the kinetochore, and there's a kinetochore on either side of this duplicated chromosome that you see here, and then some of these spindle fibers, which are microtubules, which are a cytoskeletal element, some of them will hook onto the kinetochore, and they're called kinetochore spindles or kinetochore microtubules. And then some of the spindle fibers that are called non-kinetochore spindle fibers or non-kinetochore microtubules, they will push on a spindle fiber coming from the opposite centrosome, and then they push the 2 poles of the cell apart. So some of the spindle fibers are helping to align the chromosomes and make sure that half of the chromosomes are going to end up in one cell and the other half are going to end up in the other cell. But then there are spindle fibers that are part of the process of separating the cell. So they're more a part of cytokinesis than they are mitosis. So the spindle fibers have this dual job. And once all of the chromosomes, or really technically their centromeres, are lined up along the equator, there are going to be a bunch of enzymes and molecules that come along to double check that everything at the metaphase plate is actually set up properly. Metaphase actually doesn't last that terribly long, because metaphase is this formation. And then after it's checked, it will go into anaphase. And during anaphase, the sister chromatids are separated from each other. And when you see metaphase in a real plant cell or animal cell, remember it looks different because it's the centromeres that are really pushed to the metaphase plate, and then the arms of the chromosomes are kind of flopping off to the sides. And then in anaphase, they almost invert, because once again, it's the centromere which has the kinetic cord that's being pulled, and so the arms are just following along behind. So if the arms of the chromosomes look like they're reaching for the centrosomes, then that's probably metaphase. And if the arms of the chromosomes look like they're reaching for each other, then that's probably anaphase. So look for the centromeres more than the chromosomes themselves. And telophase is the last stage of mitosis, and the nucleolus reforms. You start to see the nucleolus again. The nuclear envelope is starting to reform. The spindle fibers are falling apart. The chromosomes are uncondensing. And then the cell cycle will be complete when cytokinesis ends and telophase ends, and then the cells look like G1. And then this cell will just keep on going and living its happy cell life until it's time to divide again. All right. The cell cycle of interphase, mitosis, and cytokinesis. As I was describing earlier, there are signals that tell the cell when to go into the S phase, when to go into G2, when to go into mitosis, etc. And if anything happens to those checkpoints, then the cell could divide out of control, make a mistake with how many chromosomes end up in the daughter cells, etc. And usually that's cancer. When there are multiple breakdowns in the cell cycle control system. And we're going to look at just one example of cell cycle control. There are many. It's actually amazing how many there are, and it just adds to the fact that how we're alive is insanity. All the reactions that are taking place inside of you that are not really necessarily directed more than they happen because different electrons are attracted to different nuclei and different ion and molecule concentrations. So diffusion and the attraction between an electron and a nucleus is why I'm able to speak to you right now, which just blows my mind. Anywho, this image is from your textbook, and it shows you how the molecule cyclin and the enzyme CDK control when the cell moves from the G2 stage into mitosis. So the G2 checkpoint is the checkpoint to leave G2. And to go into mitosis. There's an M checkpoint, and that is a checkpoint to leave mitosis. And there's a G1 checkpoint, and that's a checkpoint to say it's okay to go into the S phase. So there's sort of, some of these are named for the phase that it's leaving. So the G2 checkpoint, like other checkpoints and other signals that are sent in cells or between cells, is a process called signal transduction that you will learn about when you go to college. It is in the freshman college biology courses. Eventually, probably in the next couple of years, it'll be pushed down to high school as well, because they're just learning so much about it that chapter keeps getting bigger and bigger in the textbook. So it will get pushed down to high school very soon, I am sure. But I have some pictures of what signal transduction is that we're going to look at, but it is not on the test. What you need to know for the test will be this concept right here, but I'm going to take just a moment to go off topic for just a second, but it will help you understand this picture better. So for the G2 checkpoint, the G2 checkpoint tells the cell that G2 went great and we are ready to go into mitosis. And the way this happens is a cascade of reactions occur that allow the cell to go past the G2 checkpoint, and then also allow the cell to begin mitosis. And this process of a cascade of reactions is called signal transduction. And this image here is a very general cartoon showing signal transduction. And on the far left, you see that we have all these enzymes that are turned off, and they need a trigger to turn them on. And here, in the second picture, we see that we have a molecule binding to a receptor, and then that receptor turns on one of these enzymes. And then once this enzyme is turned on, it turns on another enzyme. And then that enzyme turns on another enzyme. And then that enzyme turns on another enzyme. And this enzyme makes something happen. That is signal transduction. And this is happening inside of you constantly. And this is what I mean when I say we have this cascade of reactions. And a lot of times this is a phosphate group that's just being passed around. And another example of a real signal transduction pathway is this epidermal growth factor. So epidermal growth factor is exactly what it sounds like. It is promoting growth, and it is a cascade of reactions that turn on a gene to start producing protein to help this organism grow. And you notice that the signal's coming from outside the cell. And then this cascade of reactions make their way into the nucleus to turn on a protein. So a protein will be synthesized. And then that protein will either do its job, or that protein might be going on to be actually part of muscle, or maybe part of the cytoskeleton, a lot of different things. But the cascade of reactions can be quite, a very, very long process. So when I say cascade of reactions, it could be hundreds of reactions that occur. Anywhere from, staying in the cytoplasm to going into the nucleus, maybe going to the rough ER, etc. So when I say that this CDK and cyclin control of the cell cycle is a cascade of reactions, this picture that you see shows you one piece of the process. So the enzyme that's called CDK is a cyclin-dependent kinase. And a kinase is a general term for an enzyme that phosphorylates another enzyme or molecule. And we have a ton of kinases because that transferring phosphate groups is how we, in general, pass energy around. And this kinase, CDK, is always off. And it can be turned on by cyclin. So that kind of is like what we looked at right here, where one molecule turns another molecule on. And that's what's happening. Cyclin turns on CDK. And when cyclin turns on CDK, we call it MPF. And MPF is the functional version of the kinase. And it starts phosphorylating a lot of molecules, and it sets off a lot of different cascades of reactions. So it sets off a lot of signal transduction pathways. And that's what this picture is. It gives you a couple examples of what this functional kinase, MPF, what does it trigger? It triggers the formation of the spindle fibers. It triggers the chromosomes to start condensing. It triggers the nuclear membrane to fall apart. How does it trigger all of these? It phosphorylates 3 different molecules that then go on to phosphorylate other molecules, which then starts a cascade of reactions that will cause the nuclear envelope to fall apart. It will trigger the chromosomes to start condensing. It triggers the centrioles to start building the spindle fibers. So this one kinase is triggering a lot of cascades of reactions. There are several signal transduction pathways that are triggered by the MPF complex. And this complex is only functional when the cyclin is bound to the CDK. If cyclin is gone, then the MPF is shut off. And that's what you see in this picture. You see the cyclin binding to the CDK. It turns on MPF. Now MPF can start all of its signal transduction pathways and will continue to do so until it is turned off. And the way it's turned off is to break the cyclin apart. So this is an on and off switch that is controlled by cyclin being broken apart or cyclin being synthesized. So cyclin starts to be constructed during the S phase. And then in anaphase, cyclin is broken apart. And as cyclin is broken apart, the MPF complex is broken and doesn't function anymore because it's just the CDK, the cyclin-dependent kinase, which cannot function on its own. It is not turned on until it binds to cyclin. So this graph here shows you 2 things. One is the activity of the MPF. All right, so how is it phosphorylating the targets? If it is, then the peak increases. And then when MPF is not phosphorylating the targets, then the activity drops off because the activity is phosphorylating the targets and setting off the cascade of reactions. And then this burgundy line is showing how the concentration of cyclin changes throughout the cell cycle. And the reason they show you this graph and this picture, you may think, okay, all I need is the picture. You know, who cares about the graph? What does that do for me? And the reason they do this is because the graph came first. So somebody in the lab was measuring all of this. They have all this data, and then they have to figure out what's happening. What would explain these data? And then they make the picture. So technically, the graph was around for a much longer time before they can ever even make a picture like this. So it is important to see the graphs. And the reason it's important is to help you analyze data and make sense of data, not necessarily as just numbers or summary. Graphs are supposed to tell you something. Graphs help you answer a question. Graphs are used to give you information way more than graphs are used to summarize information, if that makes sense. Graphs, it's like building a calculator. You are building something that you will use. A lot of times in school you have a graph as an end product, when really the graphs come way earlier in the process. So if you have any questions about that, please just email me. This may have been too much information. You're really just responsible for this. If you want to understand this on a deeper level, where you can look back and go, oh yeah, we talked about that in my biology class when you're in college one day, I can explain this to you more. And we will end here. And in the second video, we will pick back up with meiosis.